Hello, welcome everyone to Podovirus Podcast. So today we have a special episode, and I might say that every time, but we're doing lots of different things all the time these days. And I heard about this exciting AI story that's also a phage picky story. So it was the obviously perfect choice of topic. And I'm so glad to have convinced José Penadés and Tiago Costa, who are professors at Imperial College London. They have been working together. I'm sure we'll hear a lot more, but they have been working in the space of mobile genetic elements, phages, specifically phage-inducible chromosomal islands or PICIs. And they were working on this, and apparently Google let them use its not yet released co-scientist tool, which is an AI tool. And they were able to use it to kind of, sounds like, rederive a body of work that they had not yet published, but have been working on for a couple of years. And giving the AI tool this research question and a little bit of background, but none of their unpublished data, they saw that the scientist was able to kind of come up with the same hypothesis that they had recently proven but not yet published. So this is the story that we want to dig into, and you might have seen a bunch of coverage of this lately. It's been on like Forbes, The Economist, BBC. So they made it to the mainstream, which doesn't happen all the time with phages. So I'm sure, you'll be able to look into that coverage too. But we wanted to kind of talking to our scientists, our phage scientists especially, but everyone who might be curious about what are these AI tools useful for and how do they actually start using them. So I'm very excited to have you both here. And of course, my co-host Joe Campbell is here too. So we'll get deep in and hopefully find out more what are the pros and cons of using this kind of tool and what are the limits right now. To start, tell us just a little bit about what were you studying when you came across this tool? How did that even come into your lap?
Okay, so for more than 20, 25 years, we have been working with these PICIs, that they are phage satellites. So they are using theoretically other phages for induction, for mobility. And it was, at the beginning, we have quite of issues because people just thought that these PICIs are defective phages. Okay, probably for more than 10 years, we were fighting just to put the PICIs as a kind of new family of mobile genetic elements as phage satellites. And probably, 10 years ago or 12 years ago, we discovered a peculiar PICI family that there were these kind of capsid-forming PICIs, or what we call CF PICIs. And they were a peculiar family because they have all the genes required to produce the capsid, the small capsids, and to package the DNA into the capsids. But they still require tails for create infective particles. This is quite unusual because the majority of all the other satellites just eject probably everything from the phage. Okay, but this is a very peculiar thing. So, and we realized that these capsid-forming PICIs were present exactly the same DNA, okay? The same element in many different species. Sometimes we put as an example one capsid-forming that was in 7 species from 5 different genera. So it was a bit surprising how these elements, we thought that probably this is a new mechanism of gene transfer, allowing these elements to go between species and even between genera. And this is the things that we have been doing in the lab for a few years to try to understand the mechanism.
Exciting. And then were you also working with Tiago's lab, or how does that come into play?
So Tiago is a structural biologist. Probably he can introduce himself later. It's quite funny because he works in another important mechanism of gene transfer. He's an expert on conjugation. And we really wanted to know how these capsid-forming PICIs, these small capsids that have the ability to package the PICI DNA inside, how they look like in terms of structural, because theoretically the genes involved in the production of the small capsids, the capsid-forming PICIs, the capsids, are related to phage proteins, but somehow they have evolved the ability to produce small capsids and just to package the PICI DNA inside. And so we start working with Tiago because he is the expert in getting all these kind of structures and things and in fact, he solved the mystery, but I think it's better to introduce himself to
Yes,
explain.
go ahead.
Yeah. Well, we solved a fraction of the mystery, so we still don't know the full story, but I think we are in the right track. Also, and this is also supported by the AI co-scientists, that they seem to agree with our experimental data and our hypotheses. Yeah. So I come from a different mechanism of gene transfer from conjugation, but I mean, my lab and Jose's lab share this this love and excitement for the how bacteria either directly transfer the DNA among themselves by conjugation, or how they use the phages to mobilize the DNA. So Jose asked me to, or we have teamed up to understand from different angles the mechanism of how this capsid forming PICIs are able to widespread in different bacterial species. So my role, or the role of my lab, was to understand at the molecular level how those CFPs are formed structurally, how these capsids are able to to take one protein, a phage protein, and change it to make this structurally very different entities. So just to go a little bit more in detail, we understood that there are some insertions of protein sequences into the capsid forming proteins that dictate the different symmetry of the capsid forming units and make them smaller, so that they can only accommodate the DNA of the CFPs and not the DNA of the phage, which sort of makes it smaller, so that they can only are able to harbor the DNA of the PICI.
Probably it's better to, we didn't say, usually the satellite, these kind of PICIs, they have 1/3 of the size of the phage genome. So it's usually these PICIs are around 10, 15 kilobases, and the helper ones are around 45. So a classical mechanism of phage interference is for the satellites to produce small capsids, where just the PICI of the satellite DNA can be packaged. Okay?
Got it.
So, these capsid forming, they all just produce small capsids. They also have a specific terminases to package the capsid, the PICI DNA, not the phage DNA. So this is kind of very specific packaging system to package the capsid forming DNA into the small capsids that are formed by proteins encoded by the satellite. That was another unique thing compared to the satellite. Okay.
And I think I saw in one of your papers that these are not considered parasites because they're not having a detrimental effect on the phage. Is that right?
Yeah, so for many years, I think it probably was in terms of selling the papers. It was nice to talk the war between the parasites, but in this case, the prototypical member of this family, we didn't see an interference with the phage reproduction. So the phage, we don't know for other capsid formings if that's the case or not. But even we published, I think, was in 2 years ago, even some phages that might suffer for inducing the PICIs. Because, for example, we published that because some of the PICIs have anti-phage systems. So even if you have some cost inducing these PICIs, if you can move them against other phages, you can also benefit. So we don't see now them as a parasitic relationship, might be. So, but we might see more, we see them more as can be more synergistic, or, depending on the scenario. They are more friends than we thought probably 15 or 20 years ago. Okay.
So what made you want to use this co-scientist AI tool, where were you when you figured out that that was an option, and why did you set it to work on this problem?
Tiago started using it for his work. Tiago, go ahead.
Yeah. So yes. So actually, there's a Fleming initiative at the college that put us in contact with the Google, and because they knew that Google was developing this new AI LLM model tailored for scientists, and they wanted to engage with the scientists so that they could understand how good the LLM model was. And so actually, initially, this was not related to PICIs. So there was the work started with my lab trying to understand a particular question in terms of conjugation that has been unknown for 70 years. So we were interested to understand how conjugation is actually initiated. Okay, and we wanted to understand the molecular determinants behind the T=0 of conjugation. So we have challenged the algorithm, and the algorithm came up with several hypotheses, 5 different hypotheses, and we realized that it would take us many months or to understand whether the hypotheses, the highest ranked hypothesis, would be correct. Okay, so Jose was on that meeting and immediately saw an opportunity there. Okay, so why we don't challenge the algorithm with the actual our experimental data that we already have in our hands, and we were about to submit, or I don't know if we were about to submit or already submitted to one of the top tier journals. Okay, so basically we turned upside down the strategy of Google. Instead of having a AI driven hypothesis that would be validated in a few months down the line, we knew already in the case of the PICIs, we already knew the mechanism behind the spreading of the CFPs in different bacterial species, and we have challenged the algorithm with that exact same question, and so it was astonishing. I mean, the one of the top hypothesis was basically a recapitulation of what we have observed in the lab.
Wow. So were you surprised? Yeah, go ahead, Jose.
He didn't say, but the output, the hypothesis that the co-scientists suggested for the conjugation, I'm not an expert there, so I'm just, he was really impressed. So that's why I said, okay, this looks very good. I need to take some advantage here, because if the hypothesis provided by the co-scientists for conjugation were crap, probably I would have been interested. But he was really impressed. I said, okay, this is looking very good. So we did this kind of, we changed the approach, and it was quite impressive. Probably the audience probably would like to know now that, so the way this capsid forming can be mobilized between different species is very simple. Okay, they can produce these capsids with the picky DNA inside. Okay, and that was a mistake we did for many years. We always thought that whatever is released after phage infection or whatever, it's an infected particle. Okay, what we realized in this paper is that the species can be induced, or even without induction, as soon as the cell slice. What is released is just the capsid picky with the packaged DNA. And outside the cell, they have the ability to take tails from different phages. Free tails. Has to be free tails, okay? And then depending on the tail, that will determine, because we know for many years that the tails in the phage biology determines the tropism. Okay? So that's it. Okay? So there were many things on the paper, okay, until we realized that it's induction or non-induction, the cell slice, okay, the capsid, the packaged DNA will be released, no tails, and then outside will be free tails. We knew for many years as well that there were many tails that are produced in excess, okay, and even during the purification, they already showed that there were more tails than infected particles or whatever. And then it's for the same capsid, depending on the tail that will bind there, that will determine the tropism, okay? So, and then one of the hypotheses that Google says is like, you need to check the possibility that these capsids can interact with different tails from different phages, okay? The system didn't talk about how that happened or that was inducted. So it was just the final picture, okay? So many things that was in the middle, it's not. So many new concepts that we put in the paper are not on the Google document. But the final thing, that was right, okay? And it was something also quite impressive, is that we knew for the genetics, and now Tiago is solving the structure, hopefully will be soon, that there were two key players in this, the ability of a capsid forming and binding to one tail or another. Is two proteins called the adapter and the connector. So if you swap the adapters and the connectors, you will swap the ability of a capsid forming to bind to some tails or others, okay? The co-scientists also suggested we should look at the adapter and the connector.
Wow.
Okay. So it was so many things that were not, in the output from the co-scientists, but the key one, the tails, that was there. Okay.
Wow.
And it was another one. So we put this, that was quite interesting, is they suggested, the system also suggested to check conjugation. That was quite funny, as a new mechanism, because conjugation is more promiscuous than transduction, theoretically, because of the tail, the narrow for a specific tail, okay? And we have started working on that as well to see if that's true or not. But that was quite interesting hypothesis as well, okay, that this system provides.
Tiago must have been excited that it chose your favorite area.
Yeah, well, it was back to the beginning, right? That was the first thing that we have tried, was to understand a bit better
as a structural biologist. Tiago loves the adapter and the connector.
Yeah.
Have dinner or lunch with, you just need to mention the adapter, because it's pretty cool that some adapters and connectors bind to one specific tail, look to be very narrow, and others have the ability to bind to several tails, okay, from different species. And he's all the time saying, very excited to see how mechanistically, probably one or two residues, that's the word you need to mention to Tiago. Connector and adapter, that's it.
I love it.
To get a little better understanding of the process. So I assume, because you hadn't done this when you started the experiments, were the hypotheses that were generated by the computer things that you had thought about and therefore you designed experiments to test them? Or did you have ideas? I mean, how were the experiments designed by you that ended up leading to the conclusions that agreed with the AI-generated ideas?
I agree with us, because the timing is very important here. Yeah. So we have been thinking a lot, okay, how this machine got the right answer, okay?
Right.
And it's very frustrating. It's very frustrating because we have the answer there and we didn't see it, okay? As a phage biologist, we know that the tail determines the tropism. Is that right? Yeah, we know that for many years, okay? Yeah. For years. So if one DNA using a tail is in many different species, it's because maybe it's used different tails, But we were biased. That's the problem we had. And I'm killing myself for that, because for many years, I always thought, and all phage biology people will think, that after infection, what you have are infective particles.
Yeah.
With the capsid and the tail. And we knew it's very strange. We have PICIs that can be induced, but we didn't get transferred. We couldn't understand this thing because we were wrong in the way we solved this thing. We were so biased, okay? We always thought that after induction or whatever it's released from a bacterium are infective particles, both for phages and both for satellites, okay? And I think that's the big thing. So Google didn't understand what's happening here, but make the most simple connection. If you are in many different species, and to go to these species, you need a tail, you are binding to different tails. That's it.
So, look, if I could just go back to the order, am I hearing you say that you had data and you weren't exactly sure how to interpret it, and then you realized that this was telling you how to interpret it?
No, we had the data for years.
Yeah.
Okay, so for example, we have the PICI we use, the capsid form is from Klebsiella. Okay?
Right.
And we induce the PICI, so this is a prophage there that is a helper prophage. Until the paper will be published, for us, a helper prophage means a phage that will provide whatever is required to produce an infective particle. Capsid and tails or just tails, okay? So we have a strain in Klebsiella 1. We induce, we show very nice capsid-forming replication, very nice packaging, no transfer. So in our mind was because the recipients we use didn't have the receptor. But we were thinking all the time that there were infective particles. Okay? And the same happened to us in E. coli. And then one day, suddenly, talking to my student, we realized that in E. coli there were several prophages. But when we induced them, we knew that the island was heavily induced, with a lot of replication, and a lot of packaging, because we can purify the capsids from the lysate. But the transfer was very low. And then he was making mutants to delete, it was, I think, 6 prophages, one by one, to see what's the inducer or whatever. And then he realized that it was one day that he removed one, eliminate the transfer, even the low transfer, but the island was still induced. And at some point we thought that maybe will be phages to induce and phages to provide the tails. Okay? And then we start connecting the dots. And it has been known for 50 years or 60 years that you can get lambda or 80 lysates, mutant in capsid, mutant in tails. You induce them, you take the lysate, you mix, and then you have infective particles. But the people that did these experiments, they never thought taking capsid from one phage and tails from another. Never. Because we all thought that everything, that's the big bias, okay? So then that day, everything started making sense.
So if you think that I was excited when we understood that the adapter and the connector are the key factors here, you should have seen Jose on that day when he cracked this hard nut. He was very excited because he knew that this was something big, something novel. Yeah.
And you did this before the AI came into play. You cracked this, but you were spending a couple years trying to get there. And then...
More than a couple.
Okay. More than a couple.
Yes.
Many years, a scary number. And so then the fact that the AI came up with that because it did not have this bias that once you release a particle that, of course, it has its tail, it has whatever it needs to be infectious. It was unbiased in that thinking. And of course, why not? Could you have this later meeting of capsid and tail outside the cell? And that's sort of what happened.
So we also, sorry, Jose, I think we need to also to clarify a bit the chronology here, because there was other angle that was in play here, which obviously we saw opportunity to patent these new elements, because because these elements are broad range, okay, which is one of the limitations on phage therapy. You have all this narrow range. These phages using the phage therapy, they either only hit a bacterial species or sometimes strains within that species. And with these chimeric particles, pretty much you tailor-made and open up you have a toolkit where you can customize the target bacteria that you want to hit for therapy or to make a diagnostic tool. So we kept this very secretive because unless we could only speak about this in public once the patent was filed. So there was no public domain. So that's why we are very reassured that AI system would never had access to this manuscript or these ideas, because they were kept in a safe box in our computers.
Also, we challenge other systems as well with the same input, okay? And none of the other systems, even related to the scientists, And even, so I think the important thing, we didn't mention this thing in the preprint. When we got the first round with the 5 hypotheses, we asked because, as I said, it's not really a real mechanism in terms of the capsids will be released outside the cells and they will, was just, okay, check if this can bind to different cells without telling you how this interaction will happen or when will happen or whatever, okay? Like make a second round. And I said, but this, challenging the system, saying, but this doesn't make any sense because how these particles are produced, will be whatever. And the reply was crap. You know what I mean? So the system don't really understand what it's proposing. It's just connecting the dots, which makes sense. Just taking this simple thing that if the cells determine the tropism, maybe it's because it's using different cells. That's it. And it was there. We all knew that, okay? So this is the type of things that for the people, when they read the paper, they will think every single person will understand very quickly because it's obvious. And, but somebody needs to tell you, I mean? So so it was quite frustrating because it was quite, few years trying to understand. We have strains and a lot of mutants and we couldn't understand why we didn't get transferred. It's because just this tailless, we said this is a new entity. We call the tailless capsid forming, the package DNA. These are the entities that are released, okay? And
Can phages, regular lytic phages, have this sort of mix and match of tail and capsid, or have you started to look?
That.
That's so cool.
If I need to bit, I will say yes.
Yeah.
Because
do they all have this
thing,
Yeah. They have these adapter connectors. Like, do many phages have those? Is that conserved?
Yeah.
Interesting.
We have asked.
Okay.
Sorry, I think the biggest difference is that when a phage infects, the capsid and the compatible tail are already there. So we think that the number of capsids that will be released probably is lower. Okay? But structurally, the capsid forming, the proteins involved are exactly the same that you have in the typical HK97, and this is the things that Tiago. But also, we think that even other myoviruses or whatever, they work in kind of similar thing. The funny thing that we are working now, and probably the people that it's good that we will say them public, because then if somebody copy us, can always get the credit. So now that there are many anti-tail, for example, anti-phage systems that block the tail formation. So when that happens, you will have capsid.
Just freely coming out.
So we really think the same will happen with phages, okay? And the phages will have the ability to inject DNA in different species by swapping tails. If these phages in the new species survive or not, this is another question, okay? We also show that the capsid formings, when they go to another species, they have mechanism to hijack other phages that are in the new species to also be mobilized and to be transferred, but mechanistically, we think that will be the case. Okay.
Yeah. So tropism is sort of like you're like narrowing, kind of getting more high resolution on what it's actually defined by, like not necessarily just the tail, but specifically this connector point, maybe a little bit.
Yeah, I mean, the true pyramid will be always determined by the tip of the tail, right? Whether it binds the surface or the receptor in the target cell. But it's true that now the neck region where the portal, the adapter, and the connectors are will have a saying in determining that trope is not by the direct binding to the surface or the receptor, but by the generating a promiscuous or a very specific structure that binds only one or several phage steps. So that's so cool. And that's what we are looking after. Well, we sort of start to understand now what is imprinted in those structures of these proteins that determine that specificity or that promiscuity in to bind only one type of tails or a set of tails. But this is for the next story. This is the next story.
Yeah. This capsid forming. So we proposed in the paper that we published in 2023 that the genes came from the HK97 phage, let's say, but they have evolved. So this is not cross-talk between the two proteins. Even they are very similar in terms of sequence. Okay, so you have the capsid, the portal, the terminases, the protease are very specific. Okay, so everything is very specific until they arrive to a point that is the connector and adapter, that each connector and adapter needs to bind to the capsid, the PICI, or the phage PICI. And then they need to bind to the same tail. So it will be a quite funny thing. You evolve something to be completely separate in terms of capsid forming things and, the proteins involved that you cannot cross-talk or you cannot, but then the tail is the same. And probably will be a competition between them as well. So it's quite interesting to know what's happening there, because the idea we have is that probably we can create synthetic PICIs with the ability to bind to multiple tails. And if that's possible, we will limit, we will solve one of the limitations for phage therapy, we can deliver DNA to multiple strains or multiple species, or, this is the pattern that Tiago was mentioning before.
Yeah. Yeah, I think this is so cool to it feels like one of the first fresh sort of phage host range discoveries that I can remember for a while. Like it's another level of looking at it as a whole. And pairing it with the AI aspect, it feels like we have this potential set of tools that it's going to be more, not less. Like we're all going to probably start using these tools. And now we can have almost like someone in a beginner's mind, like who doesn't have all the assumptions of our 100 years of history. And then now we can get their input into just physically what would be probable or possible. And now we can kind of bounce off that whole other set of knowledge, differently built than ours.
I agree. For me, something that is kind of revenge, no nice for me. So I normally I don't say these things because people said that this is not the right things to say, but I think I succeed. Or some of the things we found in the lab was because I don't read too much.
Yeah.
Okay. So I'm not biased. Okay.
I love that. Yep.
Yeah. To some extent.
You can't admit it. It's hard to admit.
I admit, and so many times, I have a very good idea in the lab, say, wow, looks so good. And then I go to med and say, somebody in 1985 thought the same. And then you have a huge admiration for these people, but I wasn't biased. That happened, for example, with the lateral and these kind of things, because I was
interesting.
Completely ignorant I always said to my students, I trust your results, because the students sometimes said, Jose, but I don't mind what people said. You have the controls, it works well. Let's interpret your results, okay? And now I was so biased. I knew too much about the satellite. And I had this thing there, the tails, it was obvious, you know what I mean? So I think that's the good advantage for this kind of systems, that there's no bias. The system, it doesn't matter if this is a phage, it's a conjugation. We always thought that the satellite should move by a phage, because that's the name. We never thought, but why not by conjugation? It's just to put a very small sequence. So it was a good revenge, in that way, that the system, gave me in that way, because I knew too much and I was so biased. We were so biased when we talked about this thing, and then, we realized. And another funny thing is that I have no idea about tails, okay? Zero. It's something that I never, ever thought about, that the tails will be something interesting at all. There was a tail, its form, the funny things were the packaging, the capsid, this is the big papers from the capsid, it's K97, the terminate, and now suddenly it looks like the tail. It's a funny thing as well, it's because, as you said, you can have now a very simple mechanism to move things between the species,
Wow. Yeah. So was Google kind of excited that you guys showed the world a nice test case? Like, it's pretty neat how well it worked. Is it really that clean cut? They must have been very excited.
Yeah, well, that's a question for them, obviously, but I think they were very pleased to have experimental evidences that that hypotheses that the system was generating were sound and solid and verifiable in the lab. I mean, there was other studies that they were initiated by hypotheses from the co-scientists that are also reported in their system preprint, but they still have to go through the peer reviewed and then publication at the end. In our case, we were a little bit way more advanced, because we had already a full story. We knew the answer, and the system was able to recapitulate.
We tried to obtain some stock options from them, but so it's like as a joke, because
what was that?
Some stock options from Google.
Oh yeah, I was gonna say they should be funding your lab.
They didn't agree with us, okay? Because I think that they were lucky and we were lucky as well, because I think this is the perfect system to test, because, as I said, everything was there. Okay, you can produce the capsid, you can produce the tails, whatever. People has done experiments in the past, mixing things, tail mutants with capsid mutants, and then you have an infected particle. So all was there, just putting the dots with being biased. So I think it was lucky for them and lucky for us,
Yeah, was definitely a very, very, very nice partnership between the both sides. There was no funding involved, so they didn't give us a penny.
Because
so
some people
we need to be
ask
very clear with that.
We are getting money from Google. We don't. We are trying, but so far,
So there was no So it was mutually benefit for both sides.
I don't know if you check the preprint, because we didn't put. Sorry, Tiago, we didn't put in the preprint, but even the system for each of the hypotheses provides key papers that the system used to arrive to that hypothesis. You think it would be nice, for example, to include that information as well for the people because they were mentioning names, and for each name, they highlight one paper that they thought was relevant for the hypothesis, and that was pretty cool as well, So it's like for each hypothesis they ranked. They were like, I think is a few papers. It was for the tail thing, for the hypothesis we were mentioning. I think they there were 15 papers that they said, okay, this is the papers they found relevant to arrive to that hypothesis. Okay,
And they were real papers.
Yeah, real
presumably.
papers, and a lot of sense.
Yeah.
Can you tell if the other AI tools that you use that weren't as effective, did they see those papers and not realize the importance of them, or did they just somehow miss them? Or I guess in trying to understand what's different and why they're giving different answers, I guess I could broadly think of two categories. One is that somehow they didn't find the right papers, or they found them and they didn't realize, weren't able to understand the importance. So can you go back and figure out if those other search engines even, or whatever learning tools or AI tools even looked at those papers?
So part of the preprint, we had to benchmark the system, the co system with other systems. Okay, so Gemini, ChatGPT, and the output from those systems are different. Okay, so the format is not the same. However, the hypotheses and the answers are there, but some they output. Well, the co-scientist outputs the references. He outputs notable scientists that have worked on that field. So this is a very comprehensive report that you obtain. However, other systems, they do not give such a comprehensive output, because they are built differently. Cosine is very. I mean, we are not the AI people. It's for still co-scientists, at least for me, it's still a black box. But the system is built differently. Okay, and is funny enough. We have published the preprint of the experimental paper in bioarchives, and then we challenged the system, the different LLMs, and some they haven't seen the preprint. They could not even find or already published our published data where the answer was written.
I think Google. No, I think what the system found the preprint, but didn't provide the right answer.
Or okay, so it's the same. He didn't provide the right answer, even though he found the paper.
But they found the preprint. I think was for one case.
Okay.
And in other systems, they also propose things that make sense. So, for example, some of the systems propose that maybe these capsid forming can take tails that have the ability to inject DNA in different species, the same tail. Okay? But, for example, in that case, that will work for this capsid forming or for any satellite, So it's not something that is specific, because at the end, the tails are very similar. So some of the hypotheses were kind of okay, but none of the other systems provide the right thing, swapping different cells, and and even some of the systems, I can't remember which one was the last one we put in the paper. They made a very nice overview of the PICIs cycle, so it was definitely had very good access to the bibliography, but I don't know to what extent, as I said before, that's a problem. Because this is the problem we have. You have a very good knowledge, then how you interpret things that, it's something new. And that's the question that some people ask to us, to what extent these are just new hypotheses or just linking, connecting the dots. And I think so far it's more connecting the dots in an unbiased way more than thinking something that is completely novel. Okay?
Yeah, if I could ask just a couple more questions. The first is, are you in any way planning to rewrite your manuscript that you hadn't shown it based on what it did? So are you, I guess, my guess would be it would most likely affect the discussion. But are you planning to rewrite it? And well, I'll let you answer that before I ask the follow-on.
So for the experimental manuscript, nothing changed. Okay, so basically what the output from Google we already was addressed in the experimental manuscript. Even in the experimental manuscript, we provide new concepts that are not in the co-scientists never mentioned. What we are doing now is like because we thought it would be nice for to share our experience. We are creating a new manuscript that is our experience with the co-scientists.
Right.
Explaining what's the input we provide, okay, and then evaluating what's the output that we got, okay?
Yeah.
And explaining, for example, the tails, this is a good thing, but this is things where, explaining this kind of frustration or why we think eco-scientist was able to arrive to that point, even this non-bias, even did they provide the picture, and we also make kind of evaluation from the rest of the hypothesis. We preprinted that manuscript as well, and we have sent the manuscript to the journal as well, to the same journal that is the experimental one, because they might be interested in having both the experimental one and this
love it
co-scientist related one.
I guess the other thing I was thinking about, and maybe this is a hard one, is if you had done this, had the output of this LLM system before you started the experiments, I guess the hard question, what would you have done differently, if anything? Would you have done different experiments or no? Okay.
Nothing changes. Nothing changes. It's just the pace. Yes. So the way that I see the system is it's like a collaborator. Okay, it's something that you can interact with. You obtain hypotheses in the end, but they are not the final truth. You have to go to the lab, run the experiments, interpret the data, and draw conclusions. The hypotheses that the system generates are not universal truth. And the scientific method does not change at all.
Right.
What it does, it puts you perhaps in the right path, right from the beginning. So if you think
I understand that the hypothesis doesn't affect that you need to do an experiment to address it. But I guess what I'm asking is, back when I used to do experiments, often if you have a hypothesis, you design an experiment to test it. And were there things... So when you said it was quicker, is that because you would have designed the right experiments?
It would be because you have not, maybe you would not fail 90% of the experiments that you have failed, but you would just fail 50%, and this would save you half of a year or 1 year of experimental work and 1 year of funding and 1 year of salaries and 1 year of taxpayers' money.
So it would have changed things in terms of it would have changed which experiments you did and didn't do, right?
Exactly.
I give you an example, coming back to the idea that we knew that we have some strains that we can induce. The island, it's been induced, it's highly packaged, and we tried to get transfer and we didn't get any transfer because there were just capsid, no tails there, So can you imagine how many recipient strains did we try? Because we thought it was a defect in the recipient strain. Okay, capsule without capsule with this, can you imagine how many? And we didn't get an answer for that, and then we moved to E. coli, the same thing, and then start deleting these things, because we never thought that these were tailless. Okay, or we never thought that yes, if they have everything to package the DNA, just need a tail, maybe they can bind to different, So it's like at the end we arrived to the right experiments, But we failed a lot of the experiments because we didn't understand what was happening,
Right.
if they give you some kind of path, and there are other hypotheses that you realize that might be relevant for the biology of the satellites, but they were not really important for the question, because can apply to many other satellites, so there was not exclusive of this family. But the type of conjugation is the same. They provide an hypothesis that is very easy to test. Something that we highlight in this second preprint is that, at least in our example, all the 5 hypotheses are very easy to test.
Yeah.
Right.
So it was, and also, as Tiago said, we had the feeling that we were talking to an expert in the field. Even, says, what do you think about this? I think you should check this thing. Okay.
Right.
And then okay, that makes sense, and definitely, at the end, we were very happy because we arrived to the conclusion, the same one. But for the other examples, for example, the one that Tiago was saying, it's a mystery for 70 years. And nobody thought about it. Okay. Can you imagine how many experiments have people did in the past trying to understand the time zero for conjugation? This is a big question. I can't believe that these guys working on conjugation don't really know that. Okay. So the system provides you an hypothesis that looks okay. It's saving a lot of time.
Right. Yeah. And I wonder if in your paper about this method, you could sort of give concrete examples of why you would get moved from 90% of the experiments not working to maybe only 50% of the experiments not working. And sort of say, we would have done, if we had known this before, we wouldn't have gone down this rabbit hole, and we wouldn't have gone down this rabbit hole. And because, I guess maybe I'm trying to think about if someone's out there trying to say, well, why am I going to do this? I think a lot of people would be attracted to know which experiments that you did, which ended up being dead ends, you think you might have just not have done if you had run this AI prior to starting the experimental work. Does that make any sense?
Yeah, it makes sense.
Yeah, it makes sense, but you tend to forget the failed experiments and keep track of the good ones, right? So I think I don't know. That's why we arguably don't publish negative results, or maybe you should. But anyway, that's another question. But yeah, I think it's very difficult to say which experiments should have been done or should have not been done or could have been done differently.
Life.
It's hard, but I guess it's a question of
this
whether
won't
that
be an
highlights
issue. But if you think to the future, this won't be an issue, because if it will be an AI-driven hypothesis, you have never run those experiments before. Okay, so in this case, because we knew the answer, we had experimental, we had the portfolio of experimenters of fail and successful. We could do that judgment. But in the future, if it's an AI-driven hypothesis, all the experiments I think that will what will make a good or a quicker discovery is how good the human will be to critically interpret the hypothesis and design an experimental setup, although the AI system already gives you some experimental experiments that you could do to test that hypothesis, and that the human will have definitely a very important role still in this process.
So I think I can answer your question. Okay, so we knew this capsid forming, I don't know for how many years. I was almost ready to publish this capsid forming PICIs in 2010. Okay, and Richard Novick told me, Jose, you can't. Richard Novick is the person that discovered the PICIs in a staff that he called SAPIs. This is the first member of the family. Let's say he's the father of the PICI. He discovered the the first element, okay? And as I said, for many years, he discovered, I think it was 1998. And for more than 10 years, 12 years, people thought were defective phages. So when we discovered this capsid forming, that was probably 2010. And he said to me, Jose, you can't publish this thing because then people will think that these are defective phages. They have everything, they have the capsid or whatever, so you can't. Okay, so we wait. So it has been for many years that we knew they exist. They kind of manual, findings. We knew that they were in many different species. We couldn't understand why a satellite want to use half of the genome to carry genes that they can hijack from the phage. So it was kind of things. We did a lot of experiments that did not transfer or whatever. And the day talking to the student, that we realized that maybe some satellites use a phage for induction and another phage for the tail. Everything makes sense. Okay? And I think it's when Tiago was mentioning before, I said, Tiago, I think we have something pretty big. And since then, everything was very quick. Very quick, because these experiments are very simple. If you have a capsid mutant, you can have the tail, you can mix. So these things even work in natural populations. So a normal phage, prophage, when it's induced, there is a lot of tails there. That if you just mix with the capsid forming, you have infected particles. Everything. We have an experiment in the paper. We have a donor for the capsid forming, a donor for the prophage, a functional one, and a recipient. And things move, So, but that was the day that we realized that maybe this is something for induction and something to provide the tail. And maybe we realize that, everything makes sense. So if you have something from Google that says, maybe this can use different tails, you're thinking, because the experiments are very easy to do, at least in our case, I don't know in other areas. So I think it's like because of the bias, and I said, it was our fault because we didn't see the big picture, But having something that is new, it's like the same in conjugation. I never thought about conjugation, okay? Tiago never thought about conjugation, and he's an expert on conjugation. There is a guy in Pasteur Institute, Eduardo Rocha. He's an expert on satellites and conjugation. He has made a lot of programs and studies about ODTs that are required for and we sent an email saying, Eduardo, have you ever thought about this? And he said, no, never. Because satellites use phages, plasmids use conjugation. So we are so biased that things that are there, we didn't realize. So I think that's the main, the power of this system, at least for us, it's no bias. Whatever has been published, they will, highlight that. Have you, and then it's up to, as Tiago said, it's up to you to decide if that makes sense or not. And, for the hypotheses they provide, were very easy to test, all of them.
I want to touch on that last point. I think the testability is really interesting. And I wonder if behind the scenes, the AI co-scientists that Google made, maybe they gave it a lot of training related to knowing what science is like so it could understand like what would be a testable hypothesis versus not. Because I don't think that's probably not, what you're getting when you talk to regular ChatGPT or other LLMs or even regular scientists. Like, I think that's like a advanced skill to think, of all the things you could do, what are the things that are the most bang for your buck and the most testable, least time in the lab? Like, you have to have a lot of background to know that. But even filtering on that like lens and giving you the hypotheses and giving you testable ones, like it's pretty interesting that you said they're also testable. And I wonder if that's kind of like built into the system. And if so, that seems like a really nice feature. Is that your view of it?
The quickest answer, I don't know. I think this is a question for the Google team. The way that, we do know the basic principles. We know that the system generates different hypotheses. They are ranked, and then they compete and they challenge each other, and they are ranked with a halo ranking, like the chess players. We know that is a very complex system because I've heard experts on AI talking about the co-scientists,
Oh.
and and they seem to be very, very surprised on the architecture of the system, and not and in particular these novel hypotheses that have that Jose was mentioned about the ORETs and that these CFPs could also go be mobilized by conjugation. This what they call it indirect assumptions or hypotheses, and that they are not imprinted directly, or they cannot readily interpret by the data that is available. Okay. So whether there is an element of reasoning in the system that would come up with this less evident hypothesis, yeah, maybe. But this is again is something for the Google people to.
The system
to
sometimes
dissect.
provides specific experiments. For example, for the connector and adapter, they said you should check by cryo-EM how these things look. For some vesicles as well, the system suggests to use liposomes. So it was quite funny thing. Even some of the hypotheses I found, even they were incorrect, they were quite funny because we included them in the paper as a negative. So, for example, one of the hypotheses was that just the capsid forming, there were proteins there that might have the ability to bind to some specific receptors without the tail. Okay. And so it's like a different mechanism of entry. You say, well, this is and we include these controls because we have experiments with no tails to show that the tail was absolutely required. Okay. So
you ruled it out.
It was some kind of suggesting experiments, and a very easy way. Okay. But I had the feeling here that the phage wall is a very good model for the system because it's just, at least the work we do in the satellite is mutant complementation. This is some cryo-EM. We don't really know in other areas,
Yeah. Wow. Well, yeah, I think we should probably let you go on with your evening and close out, but this has been so cool. I guess my last thing I wanted to end on was just, when can others use this, or and are you still going to be using it? Is it still kind of in a testers only phase, or what's that like going forward?
So the system is just under development, right? The system is not publicly available still. This is again, that's the grounds of Google AI, but the discussions that we have had with them is that there is a process for the maturation of the system until it gets publicly a release. Okay, I think the strategy would be to to incorporate different areas of science into the system and understand how robust the algorithm is, and eventually if it pans out as well as at least with phage biology, it will eventually be released to the public. I think now is still this is written in the Google's co-scientist preprint, is that there is a trust tested program, and the labs interested in testing the system can contact Google. We will keep working with them because we have a very close collaboration and partnership with them, and I believe it will be not mutual beneficial for Google and for us as it has been so far.
We are using now with some postdocs, that they are in the process, very talented postdocs. So to challenge the system with the questions they really want to address at different stages. So there are projects that are quite new, so to see how the system answers the questions and how the system's better established. So we are having some kind of, but this is a different approach. It's now asking for new hypotheses, and we will give some time.
The forward version.
Yes, the forward version.
I like your reverse version. I think, both make sense, but it's like forward and reverse genetics, like the reverse AI co-scientist. Like, it makes so much sense. Everybody else who's sitting on data that hasn't put it out there.
We have done another thing that it's quite funny. I don't know. So we work also with plasmids and the impact of transduction and plasmid mobility. Okay. So we have a paper now. Again, nobody knows about the paper. So why you never have PAC or COS sequences on plasmid? Because if, as a plasmid, you really want to move, just put a PAC or a COS, and then that's it. You will fly. Okay. So we have an answer for that. It's like in reality, plasmids don't really want to move too much, okay? Because then if there is a plasmid that moves too much, it will lose variability or whatever, okay? But this idea of the plasmids being less selfish than we thought, still not, it's a very small proportion in the literature. So far, for many years, we thought that the mobile are selfish, okay? So when you ask the system why there is no CARS or PAC sequences on plasmids, that body of literature is more important than the cooperativity or whatever. So most of the hypotheses that are pretty good are wrong because that's just based on the selfish, They do these things, the terminase will cleavage the plasmid and the plasmid will not be functional, or what's more on the thinking that was a selfish element, okay? It was something that blocked the biology of the plasmid as plasmid. So it's a quite funny thing, The thinking is really good.
Yeah.
But, because most of the literature is based on the selfish thing. You can see that from the hypothesis, okay? So the system also needs to learn probably what papers are more important, the recent one versus I don't know. So we are working there, In some, we try against the reverse for the plasmid thing. The hypotheses were pretty cool, but they were wrong. And we are working now.
Did you try the other LLM systems with that one?
No, for this one. You can do that.
That would be interesting. I guess it could be scary, but could be interesting if, depending on the question you ask, different LLMs work better.
That's a good point. And we are now having the chat, and we are telling the system, that's very cool, but you are ignoring that these are not really selfish. You can have some kind of another type of relationship with the cells. So let me know what you think if you include that possibility as well. And we are waiting, So it's quite interesting exercise.
I guess you want to be selfish, but within reason, because if you're dependent on bacteria for survival, if you get too selfish, you might kill your own host.
But for
when
many
I was
years,
a graduate student, I was working in a lab that studied this transposon TN10, and it had this thing called multi-copy inhibition, because you would naively think that it just wants to keep jumping and making more and more, but eventually that's bad for the cell.
Right.
So it has a mechanism that prevents it from makes it transposed less when they get too many in the cell. And it was so that's I was thinking about that when you were talking about it. Yes. Because again, that's another selfish DNA that you think is the best thing would be for it to jump whenever it can. But if you start jumping too much, you trash the E. coli that it's in, and then...
Correct.
Yeah.
All right.
AI is going to find Joe's thesis soon.
Well, that's not that was someone else's work.
Well, thank you all so much, and good luck with everything. And I can't wait to see your papers come out, and we'll be posting them. And can't wait to get this out. So keep it up.
Thanks a lot.
Bye. Yeah. Thank you for the invitation. Okay, bye now.
So welcome.
Bye.
Bye.